behavior. I am Dr. R. Verma from IIT Kanpur. This is week 4 of the course, and today we will talk about networks of attention. Now, control of attention, we have so far been talking about areas of the brain that are responsible for different kinds of attention processes. In today's lecture, we will actually talk about how these different attentional processes are regulated or controlled. Okay. So, the control of attention can be both goal directed, that is top down when you are looking for something like in the visual search phenomena, or it could be stimulus directed, that is bottom up, which means that it is being controlled by the stimulus properties or stimulus features. Spatial attention is controlled by a mixture of stimulus driven and goal directed mechanisms. Let us look at it. When it is controlled by the goal directed manner, the neuronal projections from the executive attentional control systems may contact neurons in the sensory specific cortical areas to influence their excitability. So, basically when you are looking for something in a region of space, what is happening is that the neuronal projections from these executive attentional control systems, we will talk about which areas are these in a bit, they contact neurons in the sensory specific cortical areas to influence their excitability. Suppose I am looking for something in this region of space, I am through the executive control areas telling the sensory areas to pay extra attention to this specific region in space. This would therefore, as I said, lead to the response in sensory areas being enhanced for prioritized stimuli at attended locations, or the response in the sensory areas to be attenuated for stimuli that are tagged as irrelevant for the current goal. For something, say for example, if I am looking for my car keys on my table and I come across say for example, some other object, let us say my wallet, I am probably not really going to be very interested in that. In the stimulus driven variety, the stimulus would itself capture the attention of the individual, and basically the flow of commands will be more bottom up. So, what will happen is this system will involve circuits from the sensory systems interacting with those in the orienting or engaging attention system. So, basically if there is something that is popping up from the visual scene, it will automatically grab my attention and it will automatically orient or engage my attention toward that specific stimulus or towards that specific feature. Now, it seems that selective attention may actually mediate the cortical excitability in the visual cortex through a network that at least contains the posterior parietal cortex, the dorsolateral cortex, and the superior prefrontal cortex and the pulvinar nucleus of the thalamus. So, these 4-5 regions are probably where the attentional network control is happening. In more general terms, it seems that the attentional control systems are involved in modulating our thoughts and actions as well as modulating sensory processing. Now, let us look at some insight. Now, some insight about these processes are already available from the neuropsychological disorders that we have discussed in the initial lectures of this week. You already know that Balint syndrome patients suffer from bilateral lesions to the posterior parietal and occipital cortices, whereas patients of unilateral spatial neglect suffer from unilateral lesions of the parietal, temporal and frontal cortices, especially in the right hemisphere. You also know that in addition to these areas, neglect may also happen as a result of damage to subcortical structures such as the Superiorcolliculus or some other parts of the thalamus. Okay. Now, Mesullam in 1981 suggested that the disorder of neglect could actually be the result of damage to the brain's attentional network rather than due to damage to a specific cortical area. Now, what could be these areas that comprise the attentional network of the brain, and is there just one attentional network or many attentional networks? Current models of attentional control propose two separate frontoparietal cortical systems that are involved in directing different attentional operations during selective attention. So, first network is the dorsal attention system and the second network is the ventral attention system. The dorsal attention system is mainly concerned with regulating spatial attention, whereas the ventral attention system is mainly responsible for non-spatial aspects of attention, such as say for example, the stimulus driven or reflexive variety of attention. Now, it seems that these two control systems definitely interact with each other and they cooperate to produce normal behavior. These interactions might be disrupted in patients with neglect, and that is why you see the pattern of deficits that they experience. Now, let us talk a little bit more in detail about this dorsal attentional network or the frontoparietal attention system. Now, Hoffinger and colleagues in 2000 and Corbetta and colleagues in 2000 employed event-related fMRI to study these attentional control networks. Let us look at some of those findings. Now, Hoffinger and colleagues used a modified spatial cueing paradigm where participants were presented with a cue and were required on some trials to orient to one half of the visual field and ignore the other. Suppose you will get a cue, maybe let us say an arrow in the middle, that tells you to focus only on the left visual field and completely ignore the right visual field. After the cue, after let us say 8 seconds, stimuli were presented to both sides of the visual field, left as well and right as well, and the participants were asked to discriminate target features and make a response. In this scenario, a goal directed attention network could actually be identified that would be engaged by the appearance of the cue and was active prior to the appearance of the target stimuli. So, how is this happening? As soon as the cue is there, the goal directed or the dorsal attentional network is now engaged. It is becoming vigilant over this entire area of the visual field, and it is basically scanning for the or waiting for preparing itself to respond to the target that is going to come there. When the participants attended and responded to the stimulus, a network of these cortical regions will show or has actually shown increased activity. These regions basically referred to as the dorsal frontoparietal attentional network. Okay. So, the region that is sort of preparing to attend this entire visual field in a goal directed manner. So, this network basically reflected the sources of attentional signals in the goal directed control of attention. How are we making this claim that this network of regions is the one that should be candidates of this dorsal frontoparietal attentional network? On the basis of 3 things. First, the identified regions of the brain were found to be activated only when the subjects were instructed to covertly attend either the right or the left visual field. So, this is basically becoming activated in response to a particular spatial location that you have to covertly attend. Second, when the targets appeared after the cue, a different pattern of activity was observed. So, this set of areas is just getting activated pre-target. Okay. So, this is something that you have to remember. Now, when participants were only passively viewing the presented cues, they are not asked to respond, then the frontoparietal brain regions that were active in the formal condition where they were actually asked to be vigilant and respond were not found to be activated. Even though the visual cortex was anyways being activated, because any stimulation you present that is visual, the analysis of visual features will anyways take place. So, this set of findings sort of led us to the conclusion that this is a network of regions that is basically responsible for voluntary or goal directed orientation of spatial attention. Now, what are these key cortical areas? The key cortical areas in the frontoparietal network include the frontal eye fields at the junction of the precentral and the superior frontal sulcus in each hemisphere, the supplementary eye fields, the intraparietal sulcus or the superior parietal lobule, and the precuneus in the posterior regions of the parietal lobe and some of the other regions. Now, if we have another look at Hoffinger and colleagues study, we will find that the cue that after the cue was presented, but before the target displays appeared, activations were also observed in visual cortical regions that were supposed to process the incoming target. So, some kind of a preparatory response is happening there. Why could this activation be? These activations are found to be spatially specific, that is dependent on the direction of spatial attention, which as you know is being modulated by the cue. This attentional priming of the sensory cortex to a specific location, to this entire visual field that you are going to attend, probably provides this preferential processing advantage to some target inputs over others, something that has also been observed in neurophysiological studies in monkeys. Now, how is this attentional priming happening? This kind of priming could only be possible if neurons in the frontoparietal network could somehow send signals either directly or indirectly to the visual cortex, which can then modulate the processing happening in this or processing to happen in those visual neurons. Okay. So, let us look at whether that actually happens or not. Now, indirect evidence for this kind of thing happening comes from patients with prefrontal cortical lesions. Knight and colleagues have found that patients with lesions to the frontal cortex actually have decreased visual evoked responses or visual evoked potentials over the entire visual cortex A, and this evidence can be taken to suggest frontal cortex indeed probably acts as a modulator that influences the processing in the visual cortex. So, in some sense the frontal cortex is modulating the processing that is going to happen in the visual cortex. It can either prepare it for activity or it can either suppress that activity in some sense. Okay. Now, more direct kind of evidence for this kind of thing happening comes from intracranial studies that were performed on monkeys. Now, what happened? We know that the frontal eye fields are located in both hemispheres in the dorsal lateral posterior portions of the prefrontal cortex. These frontal eye fields are actually involved in coordinating eye movements and gaze shifts used for orienting attention. So, overt attention, if I have to move my eyes towards a particular object or shift my gaze, this is where the frontal eye fields actually play a part. Now, stimulation of these neurons from the frontal eye fields have actually been found to produce topographically mapped saccadic eye movement. So, basically depending upon which region of the frontal eye field I will stimulate will decide which exact region in the visual space my eyes will move to. This is an important point, please note. Now, Moore and colleagues at Stanford University investigated the findings suggesting that brain mechanisms of planning eye movements and directing visual spatial attention sort of are overlapped. What did they do? They used intracortical electrical stimulation and they recorded recording techniques in monkeys, and while they were actually stimulating the frontal eye field neurons with weak currents. Okay. So, what did they find? They find that while the monkey was performing a spatial attention task, these weak stimulations that occur resulted in enhanced performance in the attention task in a very spatially specific manner. So, the advantage was not across the entire visual field, but the attention was improved for only those locations where the attended target, if the attended targets were found to be at very specific locations. And what were these specific locations? These specific locations were the intended goal location of saccadic eye movements to be produced by the weak FEF stimulations. So, for example, if you are stimulating the frontal eye field with some weak current, and this is the region in space where the saccade is going to land, what is actually being found is that this is the region which experiences enhanced processing or enhanced attention. So, basically what is happening is even before the eye has actually moved there, the covert attention is oriented at that location and is leading to some of these enhanced processing advantages. So, this kind of finding led the researchers to hypothesize that if the frontal eye field stimulation initiates both saccade preparation and visual selection, then stimulating the frontal eye fields should also produce or induce a spatial attention like modulation of the visual cortex. Okay. So, this kind of just follows from this set of results. Now, to test this hypothesis, Moore and Armstrong in 2003 placed a stimulating electrode in the frontal eye fields to allow them to deliver a very weak electrical stimulation while they were recording from the V4 neurons whose receptive fields were located in the visual field where stimulation of the FEF would direct a saccade. So, suppose this is the region, again going back to the same example, this is the region where the FEF stimulation would lead a saccade to be. This region was supposed to be lying in the area V4, in the receptive field of a V4 neuron. Now, what did they do further? They presented a stimulus to the receptive field of this V4 neuron. This stimulus could either be a preferred stimulus or the unpreferred stimulus for that neuron. Remember the red horizontal stimulus versus the green vertical stimulus. Let us suppose again that the red horizontal stimulus is the preferred stimulus. Now, the neuron's elicited response is typically always weaker for the non-preferred stimulus. Again, they then applied the stimulation, weak stimulation to the FEF site 200 to 500 milliseconds after the appearance of the visual stimulus. Okay. That is where they applied the FEF stimulation. This delay allowed the researchers to examine the effects of the FEF stimulation on the activity in the V4 neuron that was actually being evoked by the visual stimulus. Let us look at what happened. This FEF stimulation could have either of the three outcomes. Either it will amplify the V4 activity, interfered or slowed it, or had no effect on it. What is indeed found was that while the monkey was fixating on a central point on the screen, a weak stimulation of the FEF enhanced stimulus evoked V4 activity. So, if you stimulate the frontal eye field neurons, that is basically enhancing the firing that is happening in the V4 neurons in response to this visual stimulus, and obviously that happened more for the preferred over the non-preferred stimulus. So, basically what we can see is while you are stimulating this frontal eye field area, this is a way to enhance attentional processing or enhance density processing at the area where the saccade was supposed to land. Okay. Now, so if these V4 neurons were not activated by the visual stimulus, suppose it, you are talking about the non-preferred stimulus here, then the stimulation of the FEF neurons did not affect the activity of the V4 cell at all. So, if you look at this result, this mimics the ones observed when monkeys attended and ignored stimuli in V4. FEF signals appear to participate therefore in goal directed attention control over V4 activity. So, when you are actually voluntarily actively looking for something, or then basically what you can expect is the area V4 activity will be increased, or the neurons in the area V4 of the visual cortex will experience heightened activation. Now, one could ask that given that goal directed signals from the frontal cortex can modulate neuronal activity, is this modulation task specific, or it is a generic modulation of the entire areas? For instance, if the task is to identify a face, will the goal directed signals alert only the fusiform areas, or they will alert the entire visual in the entire extrastriate cortex? Now, Morishima and colleagues in 2009, they wanted to investigate and understand this question better. So, what they do? They put together a task where human participants are cued on a trial to trial basis to perform a visual discrimination task for either detection of motion direction, whether something is moving upwards or downwards for example, or face basically, or look at say for example whether it is a male face or a female face. So, gender discrimination and motion direction discrimination. So, moving stimulus and face stimulus. Now, the cue was followed by either a short interval of 150 milliseconds or a long interval of 1500 milliseconds before the stimulus was presented. So, the cue 150 milliseconds and the stimulus, the cue 1500 milliseconds and the stimulus. So, this is the experimental setup. The stimulus was actually vertical gratings that could move to the right or the left, and when superimposed of an image, superimposed on an image of a male or a female face. So, for the gratings you have to tell whether it moved left or right, for the face you have to tell whether it is male or female. For half of the trials, 134 milliseconds after the cue, the FEF was stimulated using TMS. So, you know that in the TMS method, TMS methodology, weak currents can either excite or inhibit activation in targeted areas of the brain. So, basically 134 milliseconds after the presentation of the cue, the frontal eye fields were stimulated. Now, Morishima and colleagues used TMS at low enough levels so that TMS does not really impede their performance during the task. So, basically what the TMS did was it did not really modify the processing in the FEF per se. Instead, it generated a signal in the regions of the visual cortex that were functionally interconnected with the FEF. So, basically some regions which are connected with the FEF were actually observed to show some kind of response. Now, the changes in the visual cortex activity that were measured by recording the ERPs generated by the activity of the human motion area and the face processing area. So, basically these two areas showed some activity depending upon the kind of stimulus that was presented, or whether you are attending to motion or gender of the face. These results revealed that when the participants were cued to discriminate the motion stimulus, TMS activity in the empty V5 area was increased, whereas when the participants were cued to discriminate between the gender of the face, the same TMS wave was found to increase the activity in the face processing region. So, this TMS based stimulation of frontal eye field is actually not really changing something in the frontal eye field activation per se, but what it is doing is it is in a task specific manner enhancing the activity of areas which are in some way tuned to process the upcoming stimulus. So, it is again sort of a preparatory response that you can see. So, the impulses from the FEF actually code information about what is the task that is to be performed, indicating to the dorsal system, indicating that the dorsal system is involved in generating task specific goal directed attentional control signals. Alright. So, this study therefore demonstrates that FEF has actually an influence on visual cortex. So, in some sense that this is a member of the attentional control system or the dorsal attentional control system, which modulates activity in the visual processing areas. And this goal directed influence is specific, basically meaning that functional connectivity between the FEF and the specific visual areas is increased as per the function of the specific state of attention. What is it that you are going to process, that is basically going to get enhanced. Now, let us talk about the role of the parietal cortex in control of attention. As you know, where the posterior parietal lobe is a major cortical region that is supposed to be a very important part of the frontoparietal attentional system. We have seen that damage to the parietal cortex is related to several disorders of attention such as neglect. We have also seen that the dorsal areas of the parietal cortex along the intraparietal sulcus and the superior parietal lobule belong to the dorsal network, and the ventral areas, the bottom areas which form the part of the temporoparietal junction, form part of the ventral attentional network. Okay. Now, the parietal lobe has extensive connections with the subcortical areas like the pulvinar of the thalamus, the frontal cortex, as well as some of the other parts of the visual pathways, and it also contains multiple representations of space. Now, let us talk about attentional shifts. Now, attentional shifts are correlated with significant changes in the activity of the parietal neurons. When attention is directed towards a stimulus, the firing rates of the parietal neurons are known to increase, both when the stimulus is a particular target, or say for example, you are just covertly analyzing the features of the particular target. Now, when the monkey is just waiting for the next trial, the parietal neurons are not shown to fire in response to the visual stimuli in the receptive fields. So, it is basically when you are actually attending to something or looking for something is when the parietal neurons will show activity. Now, to investigate the role of the lateral interparietal area or the LIP neurons in visuospatial attention, Beasley and Goldberg in 2006, they collected intracranial recordings of the LIP neurons specifically from the monkeys, while the monkeys performed a discrimination task. Now, the monkeys were asked to detect the properties of a stimulus at a covertly attended location to determine whether to execute a planned saccade to that location. While the animal was covertly attending that cued location, occasionally distractor stimuli could be presented at other locations. So, this is the cued location, this is where your attention has to be directed. Sometimes on some trials, some stimulus is being presented, a distractor stimulus is being presented at these uncued locations as well. Now, the LIP neural activity was compared with when there was a distraction versus when there was not a distraction, and this overall pattern of performance was also compared. So, when is the monkey performing, say for example, how good or how bad is the performance when there is distraction versus when there is not a distraction. Now, what did they find? The best performance was observed when the target feature to be discriminated occurred in the location where the LIP neuronal activity was actually higher. So, if the neuronal activity was high at the attended location, the performance was better for targets presented to the attended location. So, in the area where the LIP activity is higher and if the target comes there, it is processed better. That is one. Now, if say for example, a distractor had been presented and the LIP activity had switched to being higher at another region of the LIP, then target discrimination was found to be better at that unattended location. So, wherever the distractor is being presented, let us say that is capturing the attention in the initial few seconds. Now, more specifically, that is basically what I am coming to. More specifically, right after the distractor appeared, the probe performance was better at the location of the distractor. Okay. So, it is indeed grabbing some attention as an exogenous cue, but this pattern changes about after 400 milliseconds. So, for the remainder of the plot, what happens is the performance is found better at the cued location, at the saccade target location, as opposed to the uncued or invalid trial locations. Now, this sort of informs us that the distractor has actually been able to capture attention for the initial bit for up to let us say around 400 milliseconds, and the attention is being directed at this point, but as soon as 400 milliseconds have passed, the advantages to the cued location are restored. Okay. this is where you can see that the up till 400 milliseconds the distractor has sort of grabbed attention, there is a larger response, but post 400 milliseconds you can see that the saccade location or the attended location is basically getting you better responses. Now, by looking at the pattern of activity over the extent of the entire lateral interparietal area, the researchers could actually predict the monkey's performance. So, they could actually predict that okay, this is the region where the attention is there, this is where the performance will be better. Corollary, by corollary they could also predict where the attention is there. So, for example, if the performance is better in this region, this is probably where the attention of the monkey is momentarily captured. Now, Bisley and Goldberg interpreted that the activity in the LRP provides the monkey with a sort of saliency or a priority map, and this priority map, what it does is it pulls the different individual feature maps, so color map, location map, shape map, etc., and this sort of combines all together and comes up with a saliency map. This saliency map can then be actually used by the oculomotor system, the system that controls the movement of eyes, as a saccade goal, rather when the saccade is made to an appropriate location. So, basically depending upon where the stimulus is going to be salient, that will probably automatically become the goal of the next saccade, and that is where your attention will be oriented. So, at the same time the visual system will also use this map to determine the locus of attention, where do I have to pay attention. I look at this saliency map and I find that this is the region which is standing out, so my attention sort of automatically gets directed over there. So, it appears if you look at these results that the LIP is actually an area of the parietal cortex and a member of the dorsal attentional network, which deals with the location and the saliency of objects. At what location in space is what objects very salient, and that is what will grab your attention. Now, we have talked mainly about the dorsal attentional network. Let us talk a little bit about the ventral right hemispheric attentional network. Now, Corbett and colleagues have proposed that this ventral frontoparietal attentional network is actually is almost like a standing guard and is vigilant for any significant stimuli at any of the location across all sensory modalities. If something grabs my attention, or if there is a salient stimulus in the entire visual field, is a visual stimulus or an auditory stimulus or a tactile stimulus, anyways my attention will be grabbed by that stimulus, and the areas that will engage will be these will be the areas that we call the ventral right attentional network. So, this network, as the name suggests, is highly lateralized towards the right hemisphere. There is higher activity in the ventral regions on the right hemisphere. Now, so this network includes mainly the temporoparietal junction in the posterior parietal cortex, which is located at the juncture of the posterior parietal lobe and the inferior parietal lobe. So, we will just probably see it in a bit. Now, it also includes the ventral frontal cortex, which is made of the inferior and middle frontal gyri. So, here is where you basically can see the temporal parietal junction. Here you can see the middle temporal gyrus, here you can see the ventral frontal cortex, and so on. So, Corbett and colleagues observed that when a person selectively attends a region of space, and if a relevant stimulus appears somewhere else, more ventral areas of the brain are engaged in detecting or responding to that stimulus. These ventral areas are not activated during the generation or maintenance of attention, nor with visual search. However, the temporoparietal junction gets actually strongly activated during target detection, when a target is detected, especially when the target is appearing at an unexpected location. Interestingly, this activity at the temporal parietal junction is much stronger in the right hemisphere as compared to the left hemisphere. So, the right TPJ is shown to respond equally strongly to novel stimuli both in the right and the left visual fields. Similar regions are also found to be activated by infrequent changes in stimulus properties independently of the modality of change. So, for example, if the stimulus changes its color, its sound, its shape, any unexpected change happens in the stimulus, this is the area, the temporoparietal junction, which will get engaged, or the ventral attentional network will get engaged. So, this ventral attentional network can be activated by stimuli that are unexpected, or stimuli that change in unexpected manners, say for example like warning stimuli. It seems that this temporoparietal junction appears to act as an alerting signal that interrupts the current attentional focus of the individual. So, for example, let us imagine that you are a pilot and you are riding in the plane and you are actually sitting in the cockpit of the plane, and the flight is going on as per schedule, everything is nice, and you are supposed to be relaxed and everything is going as per plan. Now, suddenly one of the buttons or one of the lights starts blinking rapidly. Maybe it is a red colored light, maybe there is a sound that starts coming beep 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 beep, something like that. So, that is basically what is a warning signal. To detect that kind of a warning signal immediately, to counter or to understand that unexpected change in either the feature of the stimulus or the location of the stimulus, basically is going to be done by this temporoparietal junction and the entire ventral attentional network. Indeed, lesions to this temporoparietal junction has actually resulted in deficits in disengaging spatial attention. So, if this region is damaged, you will not be able to orient, disengage from the region that you are working at and orient to the new location where the warning signal is being presented. Further, Corbetta and colleagues actually suggest that the dorsal network, primarily the intraparietal sulcus, is the one that actually provides the temporal parietal junction with behaviorally relevant information about stimulus, that is the salience information, that this is where the salient stimuli are lying and you need to get oriented over there. This ventral system that we are talking about, therefore, can be said to be involved with stimulus driven attention, the detection of salient targets and the reorientation of attention. Okay. So, while you are doing this task, you are actually involved in flying the plane, everything is going as per plan. In a goal directed manner, you are monitoring some of the things that are supposed to be monitored, and then there is suddenly this warning signal. The reorientation of attention to this warning signal is what is being accomplished by this ventral attentional network. So, this is, as I just showed you, the regions that form the ventral attentional network. Now, other than the regions in the cortex, there are subcortical components also that play a very important role in the attentional control networks. One of the most important region is the superior colliculus. We have been talking about that a lot. The superior colliculus and the midbrain structures are typically involved with the control of eye movements. Now, these regions are made up of several layers of neurons that receive inputs from many sources, including the retina, other sensory systems, basal ganglia and the cerebral cortex. Now, the superior colliculus actually projects multiple outputs to the thalamus and the motor systems that control eye movements, along with performing some other functions. Now, for instance, inputs from the frontal eye fields helps generate intentional saccades, where inputs from the parietal eye fields aid in triggering reflexive saccades. So, the frontal eye fields basically help the eye movement system to generate intentional saccades, whereas the inputs from the parietal eye fields help in generating reflexive saccades, which are in response to peripheral stimulus or in a manner when the stimulus is grabbing your attention. Now, it has been shown that the superior colliculus neurons form part of an eye movement system or eye movement control system rather, that mainly has a role in overt over covert aspects of attention. So, when you have to actually move your eyes to particular target locations, this is where the neurons in the superior colliculus are actually going to play a part. And these neurons are found to be sensitive to the saliency of stimulus, and therefore, they are able to guide the movement of the eyes to regions in space that contain the salient stimulus. Patients which have degeneration of the superior colliculus and parts of basal ganglia suffer from a disease called progressive supranuclear palsy, and this disease is characterized by people having difficulty in shifting their attention and being slow to respond to cued targets. Okay. So, if there is a cued target, you will not be able to orient your attention by moving your eyes towards that location, and that is basically because the superior colliculus neurons are not functioning properly. The other region in the subcortical area, that is a part of the attentional control network, is the pulvinar nucleus of the thalamus. Now, the pulvinar nuclei is actually a group of nuclei that have connections to several parts of the brain. This region has visually responsive neurons that are selective to color, motion or orientation. In addition, this region also has, it contains a retinotopic maps of the visual world, and it has interconnections with the frontal, parietal, occipital and temporal cortical areas. So, this is a well connected region which sort of also has the retinotopic map of the visual field. Now, typically pulvinar neurons show high activity when a stimulus is the target of the saccadic eye movement, or when it is being attended even without eye movements to the target. So, if there is this target here which I want to attend, the pulvinar nuclei will show more attention, or they will show more activity. So, this structure has been found to be, or may be found to be, involved in both voluntary and reflexive orientation of attention. To test this or to investigate the function of the pulvinar region in the attentional control, Peterson and colleagues basically deactivated this region in the monkeys, and they observed the pattern of attention change. What did they find? It was found that monkeys immediately started having difficulties in orienting attention covertly to targets in the contralateral visual field. So, they could not attend to targets in the contralateral visual fields. Also, the monkeys faced difficulty in filtering out distracting information from the visual field. So, they could not control the orientation of attention. Also, it was found that when competing distractors were present in the visual field, they had difficulty discriminating the color or form of these objects. So, basically all kinds of orientation to targets is basically suffering when the pulvinar nuclei of the thalamus are damaged. Finally, patients with pulvinar lesions have been found to have deficits in attentional orienting, and they also have been found to have problems in engaging attention at a cued location. So, when you are cued, say let us say there is an arrow towards the left side or the right side, if the pulvinar nuclei are damaged, it will be difficult to orient your attention to this region. Typically, what happens is these patients with pulvinar nuclei damage show high reaction times for both valid trials, because they are not being able to orient their attention to the cued location, and also invalid trials, because they are not being able to discriminate the cued from the uncued location. If these stimuli are appearing in the contralesional visual field. Now, just something that you will need to remember is, when we are talking about deficits resulting from damage to particular areas of the cortex or particular areas of the brain, we are not really saying that, say for example, no other area is involved. Say for example, when I am saying that people who experience difficulty in orienting attention as a result of damage to the pulvinar nuclei, it does not mean that, say for example, other areas are not involved in orientation of attention. I am just saying that if this area is damaged, this is the aspect of deficit that is most typically observed. Okay. So, with that, I would like to conclude this week, conclude the chapter of attention. We have talked about various things. We have talked about disorders of attention, we have talked about specific kinds of attention, space based, object based, feature based. We have talked about attentional networks today, and this sort of completes a total idea of how is attention manifested in the brain, and how is attention basically manifested also acts as a very, very important aspect of processing information in the visual area. Thank you.